So with that, today it's my sincere pleasure to welcome today's keynote speaker, Dr. Elana Fertig, who is an ITCR PI, a second time ITCR PI. Elana is director of the Institute for Genome Sciences and a professor of medicine in the Division of Oncology at the University of Maryland, Baltimore. Her research focuses on advancing new predictive medicine paradigms for oncology by converging systems biology with translational technology development. And her computational cancer biology research is actually inspired by her background as a NASA fellow in weather prediction. And so now, as an authority in computational oncology, Elana has been a leader in establishing spatial multiomics technologies, matrix factorization, and transfer learning as now current mainstays in bioinformatics. And her transdisciplinary expertise has made her a sought-after mentor and recognized leader of new training paradigms that converge oncologists, pathologists, basic biologists, and computational investigators, as well as engineers, to advance the next generation of computationally driven oncology care. So I think Elana is just an outstanding investigator to kick us off today, and I'm really looking forward to her talk. Elana, the floor is yours.
Wonderful. Thank you so much for, having me here and the warm introduction and, just so appreciative of everything the ITCR program does for the community, and have the opportunity to be here as well as, present to trainees. So I'm going to pull up my slides now, but one of the things that I'm hoping for is that a lot of the talk and a lot of the time can be more focused on questions, for, given the nature of this meeting, as opposed to just me talking at you in terms of the research that you can read about in papers. Um, and so I'm going to be talking today about some of the work that we've been doing, thanks to our ITCR project of how do we start blending mechanistic math models with single cell and spatial data, focused on work that, is really near and dear to my heart of trying to understand the progression of pancreatic cancer, to improve early detection. So just briefly, my scientific disclosures. So before I get into the science of what we're doing, with ITCR and as a whole, I want to just talk about, in interest of this talk, of this community, of how I got here, for all the trainees. And what is IGS all about? What is the Research Institute all about? I think these were things that were all foreign to me when I left my research training. And had always expected when I went into grad school to go on a very straight linear path, ending up back in applied math department, never touching anything with biology, because I was adamantly not doing biology. And how do you kind of go on a career and what does that look like? So right now I'm the director of the Institute for Genome Sciences at the University of Maryland, Baltimore. Um, it's a group that has really humbling origins, founding out of 2007 at University of Maryland, where it moved the group, from the Institute for Genome Research, Tiger, out of the Craig Ventnor Institute to really, make the genomics program that they had from the early Human Genome Project days accessible to a medical school in the move to Maryland. So there's about 25 research faculty who are running research labs, all focused on different areas of genomics. Obviously, my lab focuses on cancer, but there's areas in aging, infectious diseases, gender and reproductive health. And so it gives us amazing opportunities to really apply these technologies at scale across the whole area of human condition. We manage a lot of large-scale data infrastructure for national genome sciences projects, particularly through NHGRI. And so we're hoping to, one of the things Julie and I have been talking about is what are the opportunities that we can take some of these techniques and bring them in to the ITCR community. And also a lot of education programs, which I know is near and dear to the ITCR community too, to scale out the genomics infrastructure. So my lab, who am I and what does my lab look like? So again, I have my PhD in applied math, originally, but now director and professor of medicine somehow, in spite of not being a biologist, by formal training. And so I think for one of the trainees, I think one of the things that I would hope you get out of my talk is, you know, learning and the ability to learn deeply doesn't end when you leave school, right? And that's one of the, I think, the powerful things about research is that we're constantly in a stage of learning about things that we don't know. And if you embrace that path and embrace the uncertainty that comes with that, it can take you in really unforeseen directions. So I'm here. Likewise, Michael Oakes, who's a PhD in physics. So Mike was my postdoc mentor who did his PhD in astronomy and then introduced me to computational biology, and now has subsequently retired into my lab as my lab manager, which has been a fantastic thing for the group. Daniel Bergman has a PhD in mathematics. He's now an assistant professor, and he's leading a lot of our work in the math modeling that I'll talk about. And Dima Livos is our PhD in bioinformatics. He, and continuing on our theme of not going in a linear path, Dima went to work for finance for 10 years, doing data infrastructure and pipeline development in finance, and now has come back to my lab to work on spatial transcriptomics analysis pipelines and data infrastructure around high throughput multiomics data. Jeanette Johnson has her PhD in immunology. She's a postdoc in my group. Kathleen Noller in bioengineering, who's a postdoc fellow. And Varun Rao is a graduate student focused on epidemiology and human genetics. So that's our team. And, we're fortunate to be supported by ITCR for a lot of the work that we're talking about today, as well as other foundations. So my obsession research-wise is how do we start combining molecular measurements that we're getting from these high throughput omics data with mathematical modeling to predict a tumor's response to treatment or how the tumor develops in time, right? So a lot of what we're getting when we get these high throughput data sets is where the tumor is right now, but how do we start moving those forward in time? And as Julie alluded to, right, I started my career in weather prediction. I apologize, this figure's a little bit blurry on my screen, but that's really always motivated how I think about this, where weather prediction, I would argue, is about 50 years ahead of where biology is in terms of how it does predictive modeling. And the way that it's able to do that type of predictive modeling is that there's large scale, high throughput data sets of the atmosphere that are available at every single time point. And there's sort of multimodal data coming in there from weather balloons and satellites that are combined with mechanistic models and now AI tools where it calibrates your simulations against real world data to go forward in time. So I've always been sort of obsessed with the concept of how do you take these types of predictive methods that are being developed for complex systems in weather and apply them to biological systems and cancer. So how did I get here? I started out with my BS in physics and mathematics from Brandeis University, and did my undergrad research in astronomy and fluid dynamics. And the research in fluid dynamics really brought my interest into atmospheric sciences, and that was what I did my PhD on at Maryland, where I had the fortune to be a NASA fellow, as Julie alluded to, and focused on weather forecasting. That focus on weather forecasting got me interested in multimodal data integration, which I got very fortunate to work on in cancer, and then start working on. I got a K-25 from the NCI to transition as a mathematician into cancer research, which really launched my career, and have been here ever since, starting to think about cell-cell communication networks. I took a more data-driven approach because it seemed like that was what was coming in, and now we're starting to pivot back to the math modeling now that the data have become more mature. So as I talked about, I think a lot of what I think about in my research is the parallels of weather prediction to cancer. How do we take multimodal data, now in this case from biological systems, really characterize where these tumors are, and then start using that to predict possible outcomes, and then ultimately get to control of tumors? So this is what we're very fortunate to have our ITCR U24 start to develop, and enable us to develop the software to be able to do this. And so again, very grateful for the consortium for going along in this relatively crazy ride. So, I'm going to talk today about the development of pancreatic cancer and how we're applying these tools in order to understand that. So, one of the challenges with pancreatic cancer is that it's diagnosed at a very late stage. And it's unfortunately, when it's diagnosed, it is tends to be late stage, it tends to be metastatic, and it's unfortunately almost as a result universally fatal for patients very quickly. And one of the things that's tricky about pancreatic cancer from a treatment perspective and a therapeutic perspective is that it has a very dense immunosuppressive microenvironment. So it has a lot of fibroblasts, it has a lot of macrophages, it has a lot of monocytes and other cell types that really block that immune response from enabling any T cells or B cells to come in and induce an immune response to those tumors. And so, one of the things that we want to try to do is understand what are the characteristics of the cell cell interactions of pancreatic tumors. And how do we understand how that evolves to figure out where the tumor is developing that immunosuppressive microenvironment? What are the characteristics that enable it to develop that? And then can we target those or even prevent the disease? So with this audience, I think it goes without saying that this, sort of understanding of cell-based biology has really been paralleled by single cell technologies. And when we have single cell data like single cell RNA-seq, we're now really able to characterize the tumor immune microenvironment. So this was work led by Ben Kinney-Koster and Jackie Zimmerman, starting in the pandemic, where we collated public domain single cell data of pancreatic tumors. And we're trying to start characterizing what are the cell types that are there and what are the type of cell plasticity that we observe in the tumors by collating public domain data. And of course, what's the first thing that you do when you get single cell data, right? You run some sort of clustering analysis, build a UMAP, annotate the cell types, everything's beautiful, and you say this is what's there in the tumor. Of course, if you were just to get to that cell type annotation, arguably, why bother with single cell data at that point as opposed to just doing flow cytometry or some other lower dimensional technology, right? These are very expensive technologies with very high dimensional measurements to just get down to cell types. So, in addition to capturing cell types, transcriptional data also captures axes of cellular identity, right? So, the clusters give you what cell types are there. But because when you're measuring all 20,000 genes in the human transcriptome, right, when you have these measurements, you get information about what are the phenotypes of the cell, where are they in evolution, where are they in cell cycle, where are they spatially, what are the cellular niches? All of these leave footprints, right, on your transcriptional data. And our goal as computational biologists is to say, can we infer from these data what types of phenotypes are there and what we observe in the data? So Genevieve Stein O'Brien realized that this notion of axes of cellular identity, right, and these different phenotypes were perfectly mirrored in the mathematics of matrix factorization. So what do I mean by that? Right? If we take away the sort of world of like tumor and normal cells, right? And we have all the single cell data, and we go away from 20,000 dimensions, and I'm going to draw here a cartoon of just four genes whose expression is on the y-axis, and on the x-axis I have each cell. You can very clearly by eye see that there's some genes and where it's predominantly going up and down in the second group of samples. Let's say those are the normals. Some group going up and down in the first. Let's say those are the tumors. And then other genes that are along for the ride that are doing both, right? Like that, for example, cell cycle could be occurring in all cells. So we want the ability to delineate that. And the way matrix factorization does this is by saying, okay, what are the patterns in the data? So I have this up and down in the first group, up and down in the second, and then assigning gene weights to all of these. So in our first ITCR round, we were lucky enough to have the support to develop our COGAPS algorithm to enable this and start seeing, can we benchmark this in order to learn patterns of epithelial cell identity in pancreatic cancer? So this was work led by Mitchell and Jeanette Johnson. And if folks want any sort of how-tos on how to matrix factorize our Nature Protocols paper, I think, provided a nice overview for the community on that. But the biological applications are in GWIN et al., and that'll be more my focus of the talk today. So when we apply this matrix factorization approach to the single cell data, just focusing on the epithelial cells, what we notice is we notice two mutually exclusive patterns in the data. One that's largely associated with cell cycle progression in a subset of the cancer cells, and the other one that's associated with more EMT and inflammatory response in a different subset of cancer cells. But interestingly, we also observed it occurring in the tumor adjacent normal cells. So our question was, what's going on, right? Like if you have these cells, what's inducing EMT and why is it occurring both in the tumor adjacent normal cells as well as the tumor cells? So this led us to hypothesize that this was probably associated with a feature in the microenvironment of the cancer. So we went back to our public domain atlas of data, and what we observed and correlated it with basically every single cell type that was there. And what we observed was that this pattern associated with EMT was highly correlated with fibroblasts and only occurred in one of our two data sets. So that one data set, it turns out, was the only one that was enriched for fibroblasts. The Steele et al. Was actually an immune enriched data set. So the question then is this a real feature, or is this some sort of a technical artifact in our data set in one of the two data sets? So for this, we were fortunate that our collaborators had developed an organoid co-culture model where you can see fibroblasts co-cultured with pancreatic cancer organoids. And when we project the RNA-seq data, these EMT-like signatures, into the bulk RNA-seq data from a panel of these organoid lines, we observe across the board that these fibroblasts induce more of an increase in EMT in the pancreatic lines. And this is regardless of their subtype, whether they're classical or basal, we see this shift in EMT. So based on these observations, this led us to the hypothesis that fibroblasts induce epithelial cell state transitions through EMT. So we have this nice little cartoon model, right, where we have fibroblasts. What we observed is that they're signaling through integrins, which I didn't talk about. So we have an ECM component. And what we hypothesize is that they're inducing more of an EMT-like phenotype in both in the tumor cells as well as the normal that induces a change in cell migration that's mutually exclusive with more proliferation. So how do we start? The next thing that we want to do is everything that I've talked about is sort of making inferences from genomics data, right? Inferring what's there. But as I talked about, my obsession is thinking like, how do we start thinking about how do we predict these forward in time? And if I go back to my physics training, right, in a physics class, you would never try to make predictions forward in time by measuring every single data point. What do? You form systems of equations that enable you to model this forward in time. Now, with AI, this might no longer be relevant, but I still like to believe there's use to some mechanisms. So, for example, if you imagine taking the analogy of a ball falling and when it's going to fall, you wouldn't do this by tracing and measuring every single data point of that ball's trajectory. You would know the force of gravity and use that to figure out when the ball is going to fall from the initial conditions. So, what we want to do is say, can we take those same types of mechanistic approaches to form time dependent equations of cells to figure out how they're going to change forward in time? So what do we need in order to get a mechanistic model? We need which variables we're tracking, what factors impact them, and what equations regulate their evolution. So historically, this has been a very complex word problem for systems biology that enables custom coding of every single one of these components for each cell-based unit that you developed. So I was talking with my friend Paul Macklin, who is my partner in crime on our ITCR U24, about how do we start scaling this if we learn these rules of how cells interact from genomics? How do we start scaling this into mechanistic mathematical models to get these types of predictive models? And Paul was like, okay, I'm working on something called a hypothesis grammar. So what does this look like? I'm going to imagine every single cell has a type, which is going to be a name, right? That's going to say what type is it, where is it located, what's its position, what's its volume, what's its cell cycle. These are all parameters of a cell. And those are going to tell it to do certain things based on what the state is and what other cells and forces in the environment are nearby to it. And so what this lets you do is say, okay, I'm going to virtualize my cells in terms of just words, like literal words, to say, okay, I have a tumor cell. Tumor cells divide unless they're near fibroblasts, in which case they become more EMT-like. And start coding those into systems of equations automatically through his system called PhysiCell. So we wanted to see, could we start applying this to these rules that we've learned of epithelial and fibroblast interactions in tumor cells? So what we started to do is say, okay, I'm going to skip past all the some of the experimental data and just go straight to what we did in modeling. In yellow, I have virtual fibroblasts. In gray, I have virtual normal cells. And what we're going to do is say, okay, using this hypothesis grammar, encode in plain words, fibroblasts induce EMT in tumor cell, fibroblasts induce EMT in normal cell. Epithelial cells will a transformed mesenchymal normal cell will transfer into a tumor cell at some rate. And if they're not adjacent to fibroblasts, then epithelial cells can start dividing. So I'm going to seed a bunch of virtual fibroblasts and virtual normal cells in a simulated petri dish, and then start to say, okay, if I evaluate it every single time, for example, this cell is near a fibroblast, it's going to evolve according to these rules with some probability. So what we have is the fibroblasts move. You can see the cells starting to translate into more mesenchymal-like tumor cells where they move. And then what's going to happen is that eventually, when they get far enough away from the fibroblasts that they're no longer seeing that, they're going to transition into cells that are able to stop moving and start dividing. What this lets us do then is we can start to do virtual experiments instead of needing to go to a petri dish, we can go to our computer dish and start asking in our virtual cell labs, what would happen if we had different densities of fibroblasts? What are the implications of that on the phenotypes that we observe in our tumor and in our tumor cells? What we can observe is on the right, if we have very low density fibroblasts, we start to see large proliferation of the tumor that's growing. We see less proliferation where we have more dense fibroblasts in the two simulations on the left, but what we can start to observe is more invasive like phenotypes where we see more of an invasive potential near the fibroblasts. So this gives us sort of the ability to do in silico experiments, but how do we start linking this to human data? So now that we have spatial transcriptomics, right, we're able to not only measure what cells are there, but where they are spatially and start annotating them directly on spatial transcriptomics samples to see what are the different cellular phenotypes. So what Daniel Bergman has now developed as a key part of our ITCR grant is a tool called the Bioinformatics Walkthrough. And the concept of this is, let's say we have our spatial transcriptomics data and we have functional readouts of where the different cells are, then can we automatically put them into these PhysiCell models and develop software that enable us to say, here are the cell types that we observe in our spatial transcriptomic data, here are the types of rules that we have, and then generate a simulation from that. So I'm going to show an example now where we have two spatial data sets of pancreatic cancer shown on the top and bottom with different fibroblast densities. And now we can start inputting those rules into these simulations and start to imagine we're in a world where we can map forward in time what the implications are on those different cellular densities on tumor cell progression directly in tissue. The other thing that's exciting about this is that we can also start doing hypothesis testing based on human tissue. So what we started to ask is say, okay, we observed in our genomics data that the signaling between fibroblasts and epithelial cells was being controlled by the ECM. What happens if we turn that off, right? And we have just direct contact. If we start to run our simulations forward in time from that, what we observe is absolutely explosive tumor growth that doesn't respect any cellular boundaries. Whereas if we have the ECM modulating this, we see more controlled tumor cell growth, enabling us to do virtual hypothesis experiments and what I like to think of as more like thought experiments, like you would do in physics, directly on the tissue to ask what if hypotheses that we can go back to the bench and test. So, what we were able to do in order to test this is say, okay, if it's direct interaction, right, of CAFs and epithelial cells, then we shouldn't see invasion if we have calf controlled media. If it's something being deposited and secreted by the fibroblasts, then we should see this type of invasive behavior resulting only in calf conditioned media. So Fatima from Laura Wood's lab had run experiments on those same organoid, on same type of organoid model in calf conditioned media from both iCalves and mycalves. And what we observed across the board was more invasion when organoids were cultured in calf conditioned media relative to controls without calf conditioned media, and that the differences in invasive potential were not sensitive to the type of media in which the calves were conditioned. So with that, I hope that I've given you a sense of sort of how we think about taking these types of spatial multiomics data and integrating them into mathematical models in order to do more predictive modeling and also simulations of virtual thought experiments and to motivate new experiments going back at the dish. Um, and with that, I'd like to thank all the members of my lab and collaborators who've contributed to this work, as well as funders and, particularly ITCR that enables us to develop all the software infrastructure to test these crazy ideas. And with that, I'd be happy to take, questions either about my talk or, career development, really whatever is going to be useful for the group here.
Thank you, Elana. That was fantastic. I know we all enjoyed that presentation. Yeah, now the floor is open for questions and interaction. Please feel free to just unmute yourselves, turn on your video, and ask questions.
Elana, I'm happy to kind of kick things off.
Please.
I found it so interesting that you went from training that was not in the biological sciences, and now, as you say, you're a professor of medicine. And in terms of making that leap into considering the subject matter expertise, what approaches did you take to learn the biology that you needed to learn to be able to ask the right questions and have the right conversations? And how deep have you needed to get into the biology? Can you talk a little bit about making that transition?
Yeah, absolutely. You know, I So I think that it's really important to understand both, and that was one of the pieces of advice I was given when I entered the field, and that was like one of the values of the training in applied math that I had. So my particular training that I had in applied math was you had to do a qualifying exam in your subject matter expertise and a qualifying exam in the mathematical expertise. So it really taught me the value of sort of going deep into the science. And I think the other thing that I got as an applied mathematician is every single one of our seminars were sort of organized around taking the same mathematical concept and having them in two different discipline areas applied. So it was always like a paired talk, but showing how the same math applied to different areas. So that got me really interested. In terms of how I learned, I got very lucky with very patient collaborators. So I would sit in a lot of lab meetings and just ask really stupid questions, right? Like people would show a Western blot, and I'd be like, I'm sorry, I don't know what a Western blot is. Can you please walk me through this experiment? and took a number of short training courses. So at the time, we didn't have all the resources that like ITCR has put out, but I was doing courses on then iTunes U, right? Like, and trying to sort of take, like, listen to basic biochemistry and cell biology, to figure that out and take courses at Cold Spring Harbor. But I would say a lot of for me, and I think this is the great thing about being a bioinformatician, is I've learned a lot. I think where I've learned the most biology has been through each data set, right? And just kind of banging each data set, and then you figure out what's known, what isn't when you go deep into bioinformatics analysis. And you can start to see, okay, these are the cell types that are known, these aren't. But it really does require a patient back and forth with biological collaborators. And I think, we have tools that make this easier now, but there's no substitute for sitting down, getting your hands dirty with data and just seeing what's there and talking to people about, oh, this can't be right because like this violates like all these years of biology. And then you discover some weird technical artifact in your data. So
Gotcha.
I'd say just a lot of conversations. Don't be afraid of stupid questions, which hopefully is also going to invite people here to speak up more. Um, but seriously, like, and nobody knows everything, and just getting very comfortable with that and being able to say, I don't know, will it makes everybody able to talk more, including the biologists and clinicians, because they're going to have the same intimidation about you computationally as well.